Today we are going to study about mutation. What is mutation? Mutation is the spontaneous change in gene or chromosomal number. And why the mutation it happens? It happens because of mutagenic agents. Now, what are the mutagenic agents? Mutagenic agents could be the harmful radiations, for example, alpha and beta radiations, or it could be the harmful chemicals, for example, mustard gas and formaldehyde. Fine. Now, there are certain variations which are, or there are certain mutations which are not inheritable. So, what are those kind of mutations which are not inheritable? The mutation that occur in our somatic cells. What are somatic cells? Somatic cells means normal body cells. For example, अगर आपके ये finger कट जाती है, तो will that mutation be inheritable? No, that mutation or that variation won't be inheritable. ठीक है? Will that variation be inheritable? No, that variation won't be inheritable. So the variation that occur in our somatic cells, then that type of variation is not inheritable, or that type of mutation is not inheritable. Whereas the inheritable variation are those that occur during the replication process in meiosis. That is, when during gamete formation, when the meiosis it happens, and during meiosis, in the first stage of meiosis, what happens? The chromosome it replicates. When the replication it happens during that process, if any mutation it occurs, then such type of mutations are inheritable mutations. Clear? Now, silent mutation. Silent mutation are those mutations which doesn't causes any impact, which doesn't causes any phenotypical change. Fine. For example, ACT. ACT is a triplet code that code for amino acid methionine. Methionine is an amino acid, and if during gamete formation, if during replication, when the process of the replication it happens during mitosis or during meiosis, that C is get replaced with G. And for instance, if this AGT also code for the same amino acid methionine, then will there be any mutation? Yes, mutation will occur, but that kind of mutation will be silent mutation, because now also the same protein molecule will form. Is this clear to all of you? That's it. Now in CIE, the example for gene mutation is sickle cell anemia. Now what happened in sickle cell anemia? The sickle cell anemia it happens because of the recessive allele. And as it happens because of the recessive allele, that means it is able to express itself in homozygous recessive condition. Clear? Now, what happened in sickle cell anemia? For example, it's just an example, it's not the real coding for the normal hemoglobin. Fine? Let's suppose this is the coding that the DNA it codes for the normal hemoglobin. AGG, CTT, CAT, and then CTT. Now, this CTT it codes for the amino acid glutamic acid. Fine? When the glutamic acid it is present, then the hemoglobin that is formed is normal. Fine? Now, during the replication process in meiosis, sometimes this T, only one nitrogen base, it get replaced with A. Fine, during the replication process, what happened? This T, it get replaced with A. Now this CAT will code for amino acid valine, not for the glutamic acid. So now the protein that is formed due to this coding would be HBS. HBS is a recessive allele which codes for sickle cell anemia. If this recessive allele is present in homozygous condition, then this allele, this genotype will code for a person that person would be sickle cell anemic. For example, if the two carrier parents, they crossed with one another, then we know that after forming this genetic diagram, we will find that there are 25% chances of having a sickle cell anemic child. Now what happened in sickle cell anemia? In the HbN, that is when there is a coding for the normal hemoglobin, when its phenotype is normal hemoglobin, what happened? this hemoglobin is soluble. When this hemoglobin is soluble, then it keeps the RBC in its biconcave disc-like structure. But when the person is sickle cell anemic, then in that person, the hemoglobin, it get insoluble. Now, when the hemoglobin, it get insoluble, and when this person is subjected to low oxygen concentration, for example, that person is sickle cell anemic. Now, when this person is subjected to low oxygen concentration, it will be having insoluble hemoglobin. When this person is subjected to low oxygen concentration, then their hemoglobin get precipitated. When it get precipitated, it get insoluble. When the hemoglobin get insoluble in the HbS person or the sickle cell anemic person, when they are subjected to low oxygen concentration. In the normal oxygen concentration, there won't be any problem. But in low oxygen concentration, that is when the person is exercising or when the person is doing some strenuous work, then what happens? Their hemoglobin get precipitated because they are having HbS genotype. Fine. When their hemoglobin get precipitated, then it pulls the RBC inside, and when it pulls the RBC inside, then the RBC gets sickle shaped. When this RBC gets sickle shaped, then as you can see, it is a blood capillary. When these RBC, it flows in the blood capillaries, then it get entangled in the walls of the blood capillaries, due to which there will be reduced blood flow. Cells get starved of oxygen and the person get pale. The tendency to carry oxygen by this red blood cell will reduce. Why? Because the hemoglobin because the surface area to carry oxygen will get reduced. Fine. Paleness will occur, fatigue will occur. So all these are the symptoms for sickle cell anemia. Anemia means deficiency of red blood cell. How the deficiency of the red blood cell it occur? That instead of having a biconcave disc-like structure, the RBC, it gets sickle shaped due to its oxygen carrying capacity will reduce. Another example of mutation which is included in CAI is Down syndrome. Down syndrome is an example of chromosomal mutation. Now what happened in this Down syndrome? The Down syndrome, it happens when the 21st chromosome of the mother, it fails to separate during gamete formation. What happened in meiosis? All the chromosomes, they get segregated. Okay? Except for what happened in the Down syndrome person, their mother's 21st chromosome, it fails to separate. When the 21st chromosome, it fails to separate, and when it fuses with the male's 21st chromosome, then in the resultant offspring, the 21st chromosome will be having one extra chromosome. So in this way, total number of chromosome in each and every cell of a Down syndrome person would be 47 rather than 46. Now, what are the characteristic facial features of a Down syndrome person? They have flat face, flat head, bridge is not present in their nose, slant eyes, they have poor cognitive skills. They are, for example, we have lot of creases in our palm, but they don't have, they have only one or two creases in their palm. Fine, they have short fingers, broad palm. All these things are the characteristic features of a Down syndrome person.